Hello, everyone. Welcome to today's live broadcast, Assembly and Analysis of Non-Model Organism Transcriptomes using DNASTAR software. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by DNASTAR. DNASTAR is a global software company headquartered in Madison, Wisconsin that has been meeting the needs of life scientists for 25 years. We grew out of the E. coli laboratory of Dr. Fred Blattner, professor of genetics at the University of Wisconsin in 1984. Our early products reflected this background and association, and we built a strong reputation during the Sanger sequencing years of providing outstanding desktop software for DNA and protein sequence assembly and analysis. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answer is always welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing your comments and questions in the box. We'll try to get to as many as we can, and we'll follow up if we don't have time today. If you want a better look, you can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, please let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Matthew Kaiser. Matthew Kaiser is Senior Manager, NGS Applications for DNASTAR, where he has helped scientists address their sequence assembly and analysis challenges for the past 7 years. Matt's sole focus at DNASTAR is supporting customers in next-gen sequencing applications using DNASTAR's broad software toolset. Matt helps customers with their templated and de novo assembly projects, transcriptomes, exomes, metagenomic assemblies, RNA-Seq, ChIP-seq, and numerous other unique experiments. Matt has helped hundreds of scientists optimize their workflows using DNASTAR's next-gen software solutions. He has also spoken at numerous conferences and workshops regarding the capabilities of DNASTAR's next-gen software tools. I'll now turn the presentation over to Matt.
Thank you, Brenda, and thank you for joining me this afternoon. Today our topic will be assembly and analysis of non-model organism transcriptomes using some new features in DNASTAR software. I'm excited to introduce this new workflow. There's some novel new algorithms and some novel new analysis methods that we provide downstream in our software. And I'd like to introduce the company as well. I have a few slides here to give you some background in DNASTAR, and of course I'll be around after the presentation to answer any questions that you may have. So DNASTAR has software in 3 main areas: molecular biology and genomics and structural biology. My role at the company has been mostly to focus in the genomics area, and you'll see that there's a wide variety of different types of genomics workflows that we can support, and we'll describe some of those in detail. We'll really investigate this new workflow that was developed earlier this year. Our company was founded by Dr. Fred Blattner and John Schrader out of the University of Wisconsin. We've been in business for over 30 years. Our software was developed first to help support efforts with the E. coli K12 genome. Dr. Blattner was a pioneer, genomics pioneer, and was the first to sequence a bacterial genome. Our software is in many different countries. It's worldwide. It's used at academic institutions, governments, healthcare, commercial customers. We have site licenses, we have institution licenses, and individual licenses for researchers. Again, a very flexible series of options for how you can gain access to the software, and I'll describe some of that in detail at the end of the talk. Our support is worldwide as well. Our main office hours are Central Time, 7 a.m. to 6 p.m. What's really great about DNASTAR is that you can call in and speak with a representative. We can chat. Myself, I work with customers on their data set, so we exchange data sets often, and I can help you work through problems or any questions that you have. So it's really the top-notch level of support is one of the key aspects to DNASTAR. We also have additional global locations and distributors throughout the world. So we do software for researchers, and our goal is to provide extremely powerful solutions in an easy-to-use format and make them accessible either through a desktop computer, or more recently we have some cloud options as well. And so that's something that we continue to strive for, even as data sets get much larger. And of course, when NGS hit the scene several years ago, there was a great challenge because the amount of data produced sometimes exceeded what a typical desktop computer could handle. What we're going to be doing today is looking at a workflow where we really tackled a large problem, and that's a large de novo assembly of transcriptomes on desktop computers. So it's really quite a feat to handle these large data sets on regular computers. Because we've had research software for many years now, we've got over 50,000 publications. We're the most cited bioinformatics software company that's out there. So all the way back to the 1980s, and you can see it's an increasing trend. So we have more and more usage worldwide, and in a larger and larger user base. So to really sum LaserGene up, we provide all the software tools that we can to cover the three areas: molecular biology, genomics, and structural biology. And we do our best to provide excellent tools in all areas. And the tools look different, in different ways. We have the molecular biology that include things like phylogenetic analysis, multiple sequence alignment. We have sequence editors, so things that can support features, primer design, sequence editing, multiple different ways, circular maps and linear maps to visualize the data. So kind of the nuts and bolts of sequence analysis. We have a whole suite of tools that can perform all those functions. We also have structural biology that is relatively new. It's in our newest format. It expands on some of our protein analysis and does things like protein modeling and prediction of structure. So really an excellent set of tools. We're going to have new tools that really expand on these capabilities released later this year. So if you are interested in structural biology, please keep an eye on DNASTAR and check out some of the tools that we have. And of course, genomics, that's what we're going to focus on today. Genomics covers a wide range of different topics. And really, I'm going to talk about two different workflows that kind of combine for transcriptomics, and you'll be able to get an idea of what our tools can do, even with challenging organisms. And that's really kind of a nice niche for us, is we have flexible enough tools that we can accommodate folks that aren't working in the main model organisms. If you may not have a reference genome to align to, you can start from scratch with our software and build your own reference sequences and do your analysis. And so it makes it a really exciting workflow. So really any kind of a project, de novo transcriptome, the RNA-Seq, to exomes and genomes, and multiple genome alignment and comparison, and all the different sequencing platforms. And of course, Illumina and Ion Torrent and PacBio, we're working to be compatible with nanopore technologies. So we're working hard there to have some great solutions for the long reads that are starting to hit the market. And our hardware requirements, again, if you're going to try out the software, you may have some questions whether or not the hardware that you have in place is sufficient. For these larger projects like de novo transcriptome assemblies, my recommendation is to have it could be a laptop or a desktop computer, but what's really important is to have a extra space for this, what I call is a scratch disk. And you really want to have that extra space to handle the temporary files that are generated during the assembly. And on my laptop, I just plug in a scratch disk that's a couple terabytes in size, and that can handle the vast majority of exomes and transcriptomes and RNA-Seq projects that I do. And of course, if you're doing a lot of data, another drive for storage. If you're generating hundreds of gigs or terabytes of data, you're going to want to have a adequate storage drive. But nowadays computers are so fast and they have multiple cores and lots of RAM. Even your budget desktop computer can handle even the most demanding type of projects. Another option, especially if you're trying the software out, you may not have any hardware, or at your institute you might not be able to get updates more than every 5 years or so, which is more common than you would think. We also have the ability to run the assemblies up on the Amazon cloud. And so what this entails is you still set everything up locally. You take your data, you set up the assembly, you set your parameters, but when you really hit the assemble button, what's happening is the data is getting zipped up and encrypted and uploaded to the Amazon cloud, and then a virtual computer is spun up. And you use Amazon's hardware then to run the assembly, and then of course you can just download it locally to do the analysis. There's some advantages to the cloud. One advantage is that you can do a whole bunch of assemblies in a very short amount of time. And so, for instance, if you've got 10 exomes and you need them done by tomorrow, that might be difficult to do on a single desktop computer, but I can send those 10 jobs up easily and run them simultaneously using the cloud resources. The other big advantage is that you can easily share data. You can have colleagues from all over the world log in, access the data, upload data to you, and bring the data back down. So there's some real conveniences to the cloud, and of course it's secure. If your computer blows up one day, you still have your data up on Amazon. So if you're interested with cloud, there's more information on our website, and a sales rep certainly can give you pricing and additional information. So now the focus at DNASTAR has really been, really in the last 2 to 3 years, has been to make our software as accurate as is possible. And there's lots of different ways to measure accuracy, but in this particular case, the accuracy is measured via SNP calling and detecting known variants in a reference data set. And surprisingly, those reference data sets really haven't been available until just in the last 3 years or so. And these are human data sets. They're available at the NIH, the Genome in a Bottle Consortium, where a large portion of this particular human genome has been sequenced using multiple different technologies, and the SNPs are verified. So you can check the answer essentially. Take software like DNASTAR, get a data set for that genome, align it, and check. Do you find all the SNPs that you're supposed to in this known answer? And when we do those kind of analyses, and we have a workflow, and that's a whole different webinar, we won't go down that path now, but we have that workflow built into the software, so as long as you just download that data set and assemble it, we'll calculate for you, using maybe your own capture technique and your own sequencing core, are you getting 99.7% accurate? And so it's a validation workflow that we built into our software so you can validate your own process. And so what's nice about this is the same assembly and SNP caller and gapping and is used for all the different organisms. So if you're working in a non-model, you know that this is very, very accurate software, and it's going to do a very, very good job assembling that data. And some of the results here, this is just from our website, but you can see that there's consistent performance. This is our 3 human exomes, and it's this NA12878 exome. This is the answer, essentially, that we're checking against. And when we look at exome data from Garvan Institute, Eric Labs, Mount Sinai, we see that the sensitivity in those cases is always over 99%. And for some of the data sets, like Mount Sinai, it's over 99.9%. And the assembly time, a couple of hours, again, on a desktop computer, lots of data. So we have a very fast assembly time, very accurate sensitivity when detecting these known variations. So those same algorithms then that take sequence data and align it to a reference, we use those for our other workflows. Now the de novo transcriptome is actually a brand new algorithm that can take massive amounts of data and de novo assemble through a series of steps. It will de novo assemble that data and then compare it to a reference database and pull an annotation. So it's kind of a multi-step workflow. And then, of course, we can proceed on to RNA-Seq. So we're going to look at 2 workflows that are related. First, we're going to de novo assemble our transcriptome. Then we're going to use our assembled annotated transcripts and use those as references then to do some gene expression quantitation with different sets. So it's kind of a hand-in-hand workflow, but it is multiple step, and we try to automate as much of that as possible. So some of the challenges with de novo transcriptome assemblies, if you've done these at all, you know that with Illumina data in particular, if you have high-seq Illumina, you're getting millions of reads. And oftentimes it's hundreds of millions of sequence reads. And most of the de novo algorithms that are available are memory limited. So what that means is you usually have to get access to a big Linux cluster that can be very expensive and run that for hours, if not days, to do a de novo assembly. So it can be very costly. Then if you have to rerun, change parameters, it becomes a time-consuming and expensive ordeal. And then the output is however many different mRNA isoforms are present, you'll get a contig for each one of those isoforms. So you have potentially tens of thousands of contigs that are unannotated that come out the assembler, and you have to do something with those. Oftentimes you want to figure out what they are, so you want to annotate them. If your organism is novel, there may be limited annotations just for any one particular reference organism. You may want to draw annotations from multiple organisms. So it becomes very daunting working with thousands of contigs to apply annotations and then turn around and make use of those annotations. There's also, sometimes you may need to edit individual transcripts that are important to you. You may need to piece them together. So this new workflow provides all the different, it automates the more difficult steps and gives the user the ability and some flexibility to work with the output. So some of the benchmarks, now this is pretty early benchmarking, and we'll be adding more data here. This is for a particular data set. And you can see that there's a couple things. One is the assembly time versus input reads. Maybe I can put an arrow on here. So it's a little bit small. So number of input reads. So these data sets, the smaller ones have about 50 million reads. The bigger ones can have closer to 500 million, half billion reads. There are some that are even bigger. I've seen data sets with over 1 billion reads in them. And so with this new algorithm, our assembly time is it's a fairly linear relationship. And so, roughly 12 hours per 100 million reads for this data set. Some data sets take a little longer. They may be more complex or have things in them that are more difficult, like ribosomal RNA that hasn't been removed completely. So it makes the clustering a little bit more complicated. But they're generally, a rough rule of thumb is about, 12 hours per 100 million. The number of transcripts generated, you can see that at about 100 million as well, it starts to diminish. So for many data sets, you get most of the transcripts with 100 million reads. And what's happening is there's enough coverage on most of the expressed genes with 100 million reads that pretty much everything that's there de novo assembles. As you dump more data in, you do pick up some more transcripts. Sometimes it's fragments of transcripts or rare isoforms, or sometimes it's some other kind of a contamination. But for most folks, around 100 million reads, you generally get nice coverage of your transcriptome, and you can see that it really does plateau at about 100 million reads. So it's just something to keep in mind as you're generating data. You may not need an excess of data to get all the coverage that you actually need or require. So the particular data set that we're going to look at today, it's a Nile tilapia. I randomly selected this off of the short read archive. You can see the study here is up on the SRA, so you can download this data. And this particular data set has its Illumina HiSeq 2000 runs, 6.5 gigabases of data, and it has 3 different samples from different tissue types. So just a run-of-the-mill kind of a RNA-Seq transcriptome data set. And the organism does not have a sequenced genome that's well annotated, right? So it has a limited amount of annotated transcripts. So it's a typical kind of organism someone might want to do a de novo transcriptome assembly with. So the software then, to start out, it's our SeqMan engine. We call this a wizard. And the wizard is a starting point then for launching the NGS assemblies. And you can see here that we have different options: assemble on a local computer, rerun a local assembly, or assemble on the DNASTAR cloud. And at any one of these points, assemble on a local computer, it just proceeds to the next step in the wizard. And there's a next button down here that allows me to advance. If I assemble on the cloud, it'll ask for credentials to check, oh, does this person have a cloud account? Yes. Then you proceed to set up the assembly. The next option is to choose the assembly workflow. And the assembly workflow, then, you can see there's DNA-Seq and genotyping and transcriptomics and some other workflows. And as you make decisions here, it really starts to go down kind of a tree of different parameters that are optimal for these different types of workflows. So it's if we pick transcriptomics and then click on Next here. You can see that there are different assembly types for transcriptomics. I have a templated assembly or a de novo assembly. This is going to be a de novo assembly and clustering. And we get some system information then as well. And so the system information then tells me how much RAM I have on this computer, the temporary files, where all that's going to be processed, and how much space is on that disk. So again, if you're doing a demo, what's really important is that you've got enough free space to handle all the temporary files. If that says 100 megabytes, it's probably not going to work for you. It needs to be a sizable amount of free space in relation to how big your data set is. Then we just input the sequence files. So we can load some FASTQ files here. There can be one or multiple sets, and they're typically paired end Illumina. The pull down up at the top allows me to select a particular read technology. And there are enough differences in the read technologies in terms of how much data, the error profile, the length of reads, that different parameters are optimal for Ion Torrent, say, versus Illumina. And so our software is flexible enough then to accommodate the different types of input data that you might have. And so it's important to make that specification. There are some parameters, too, that can be adjusted. With this de novo algorithm, it's really a limited set of parameters. We have other workflows that have quite a few more knobs and dials that you can adjust to improve the assembly. In this case, there's really a mer size and a limit on the number of reads. And if you don't know what any of those things mean, there's a help menu down at the bottom, help button down at the bottom, and the help then is contextual. So if I click help on this page, it'll tell me what these different things are. And you can also see in the side panel, we get kind of a history. I can click on this side panel too and move back and forth through the wizard, something that's newer in some of the newer versions of SeqMan Engine. And so really limited parameters, and then I can just name what my output's going to be. This is a tilapia and where it's going to go, and my assembly output will be a .transcriptome folder. It's really a whole collection of assembled mRNAs and little editable project files, and then a table allows me to navigate. We put those all in a folder together, as well as the FASTA files of all the transcripts that have been assembled. And so at this point, it's ready to assemble, and you just click Next, and the assembly runs. And depending on how much data you have, it's going to take anywhere from probably 6 hours to a couple of days if it's a really large data set. Now, the results from this particular data set, there's about 60 million input reads, and most of them assembled. A lot of times with de novo transcriptomes, you don't get 80 or 90% of the data assembling. A lot of transcriptome data sets have genomic contamination or fragmentation, and aren't quite as clean, and you may get sometimes as low as 50 to 60% of the data. But on a real clean data set, you'll get upwards of 80 to 90%. This one took a little longer at 13 hours. A lot of times a data set this size will go 8 hours or so, and we get 27,000 transcripts that are annotated. And so what that means is those are transcripts that when the algorithm goes through the assembled transcripts and compares them to a database sequence, that it can identify them. And so the database sequence then is the mRNA. It'll automatically link up with the mRNA database at NCBI and scan the assembled transcripts against all the different annotated transcripts and find the strongest hits and say, it looks like this transcript belongs to this gene and it came from this organism. So with the tilapia, most of the assembled transcripts were identified. Some of them are considered novel. They really didn't hit anything strongly. Right, so we have this kind of collection of unannotated transcripts as well. And so it's a real streamlined kind of workflow, and the output then is an interactive report. And this report then is, I'm going to grab a different kind of a highlighter here. You can see that there's identified transcripts tab and a novel transcripts tab on this table. And there's a number of different columns. And so there's a gene name, there's an organism name, and a session number, a description is retrieved, and then there's some metrics on how well my de novo transcript matched the database mRNA sequence. So I get the length of the transcript, the match here, and I like looking at the match. And when I see matches that are over 99%, I'm pretty confident that my assembled transcript is matching, or I have most of it. It's nearly, if not complete, it's really close to being a full-length transcript. So the more that I get that are above 90%, I'm confident that I really get a nice transcriptome assembly. And there's some other metrics like identity and bit score that are more statistical, but I like to just look at how well these transcripts match the database. And so this is tremendously useful then. I can do a number of things with this table. It's interactive. I can double click on any one of these rows and go to the actual assembled cluster of reads for that transcript. And so if you want to go in and edit, you can go into the software then and edit a particular transcript. And if two contigs hit the same mRNA sequence, for instance, it's in some cases if you have thinner coverage on a transcript, you may find that you're able to de novo assemble the front part of a transcript and the back part, but they don't connect in the middle, so you get two pieces. And those are extremely troublesome when you're trying to annotate a transcriptome, because they're in there twice, and there could be thousands of those in some data sets. And so it's extremely laborious to go through and figure out where all the duplicates are. And so what we do here is we group them, and you can see this kind of a crazy color scheme. And the grouping is saying that these hit the same, in this case, the same gene, right? They have the same gene name, so we're going to group them together, and we're going to treat them that way. So if I choose my next step then, which may be RNA-Seq, our algorithm will say, oh, those are actually the same, even though they're in 2 or 3 pieces, they're really the same gene. So let's calculate an expression signal and compute them together. Rather than force the customer to go through thousands of transcripts and piece them together manually, which could literally be days or weeks of manual curation. It saves a huge amount of effort. You can still get the expression values that you need and use these transcripts, even though they're not all put together. Some of them might not be put together. You can still use them for your RNA-Seq analysis. So we can skip right to that next step, which can be RNA-Seq. And I'll just show you the interface then, which is going to look very similar. But the RNA-Seq then, I can select a transcriptome RNA-Seq, same kind of workflow. And you can see the interface will look a little bit different downstream a little bit, but I can pick templated assembly now instead of de novo. I'm going to be aligning now to that set of reference transcripts that I created. And so now it asks for the input of the template files. With the de novo assembly, of course, there were no templates. And in this case, we're loading the output from our de novo assembly. So there's a couple ways I can do this. You can see the file pathway there. It is my .transcriptome package that was the output. And the algorithm will automatically look inside that output and identify and find the files that contain the identified annotated transcripts and the novel transcripts. So it'll just kind of do that for you. Or I can just add, individual FASTA files from that output. So there's a couple of different ways I can set this up. But I'm going to use those now as a template. And I'm going to align additional data for my RNA-Seq. And so in this case, it's a multiple sample RNA-Seq. So I've loaded 6 data files. And there's some controls at the top, so you can see where it says multiple sample data. When I select that, I'm then asked to name the different groups, and I'm going to run these as separate assemblies, and I may or may not have replicates for the assembly. And so if you've got, you may have technical replicates that you want to run, or any kind of replication to normalize the signal, that's all possible. This particular data set, I do not have replicates. I have an experiment that I've labeled eye, liver, and muscle. So again, this is 3 different tissue types from the tilapia. And we're going to align these. These are paired Illumina files. So it's 1 set of pairs per sample. And we're going to align that to the 27,000 plus some novel transcripts reference sequences all at one time. And so once that assembly is done, I can open this up in our RNA-STAR software. And what we're looking at now is a gene table. And the gene table has multiple annotation fields now. It's pulling that annotation information in from our annotations that we got from our de novo transcripts. And so this is really the exciting part now, is actually making use of all those transcripts and making use of those annotations. So things like gene name, the description of the gene, the organism name, all in the header here. This all comes from automatically from our de novo transcriptome workflow. And then way over here on the side, we can see some of the RPKM values. So for the 3 different sets, we have RPKM values of the eye, the liver, the muscle. And RNA-STAR then has a whole series of tools then that I can use to compare these different sets, and that's really, you know, more of an interactive software thing. So like we do live webinars or one-on-one webinars with customers. We can show you that interactivity with the software. But once I have these annotations, I can also use them to filter and say, only show me from this type of organism or this kind of a gene. Show me, you know, genes with interesting expression patterns. And so RNA-STAR then gives us all different views. So I'm just showing here is you know, a full set of tools for comparing different sets. We have scatter plots and heat maps, and there's quick filters and statistics that we can apply. You can see that there's clustering and different tables for genes that we can make these comparisons. So it's really an excellent set of tools for making gene expression comparisons. So with that, you know, I'd like to just summarize a few things, and I said I would get back to licensing options. Again, if you already have access to the software, you know, this is just some new information for you. But if you don't have access, there's different types of licenses. We have desktop and network and institute-wide. We have cloud workstations. Feel free to contact me with any questions that you have about either the function of the software or the way that you can access it. Again, technical support is available Central Time, 7 a.m. to 6 p.m. We have many different training resources on our website, webinars, videos, tutorials, and manuals. And of course, you can always reach out to me. I can help you out with any questions that you have. And with that, I'll look and see what kind of questions we have and try to answer them on the fly. So I have one question. Can I use different annotation databases? Yes, there's flexibility in the database, and that's something, it's a little bit technical depending on the database that you have, but yeah, you can use different types of annotation databases for the workflow. And I have another question: is there a limit to the total number of sequences that can be de novo assembled? It's really only a limit to your computer resources. So most computers that have a couple terabytes of scratch disk space and say 32 gigs of RAM, we've tested up to about 500 million reads, but there's no reason that we can't do 1 billion reads or even more than that if you have the time and patience to, you know, wait a few more days for the assembly to complete. So again, with that, I'd like to thank everybody, and hope to see you next time. Thank you.
What happened? Sorry about that. Little technical issue. Sorry about that. Thank you, Matt, for bringing that important information to us. I also want to thank your company, DNASTAR, for making today's webcast possible. Attendees today will receive an email when this webcast is available for on-demand replay. We would invite you to share that email with any colleagues who were not able to log on to the live broadcast today. Thank you again for joining us. We hope to see you again next time.